morning. I hope you have a good, enjoyable spring break last week. Okay, so let's start this week 8. Can you hear me? This microphone? Okay. Okay. So this week we are going to discuss several topics. So we will continue a classification problem. So we will finish off with support vector machine. This is a large group of the classification algorithm widely used in machine learning. And we will also learn a feature mapping function, the concept, and also the kernel operation. So people have been using these strategies in high, so nonlinear classification problems. If you have data, it's very hard to classify using the linear function, but you can use the kernel transformation or operation to transform, to map this original data points to a new space. In the new space, they become separable. So this is a large group of strategy people use a lot in associated with the support vector machine learning, especially. Then we will spend the time on cell-cell interaction analysis. So this is a really kind of analysis utilize the advantage of single-cell data. So if someone argue about, so I can do differential expression analysis using bulk RNA-seq and the single-cell RNA-seq, not much difference. But you can see, you can tell them using single-cell RNA-seq, you can do cell-cell interaction analysis, which you cannot do using bulk RNA-seq, right? So this is a kind of a justification, one of the justifications why we should use single-cell data to understand your biological systems. Just before I forgot, I want to show you the neural network representation of this. I found it very intriguing to show how the neural network works. So this is like 2 layers of neural network, and the information is an image has been using probably CNN convolutional neural network framework to read that information, compress that into a vector. And this vector, I haven't count how many neurons this vector is using. And then each neuron will process the information, aggregate, and generate output and send it to next level of neurons. So in this video, we show the values of each of this vector weight, okay? Each of these neurons, how the weight value is, and at the end, it finish off this classification problem and to recognize what you write in this panel is which character, which digital. And that's involved all this linear algebra operations. It's all about vector multiplications between 2 vectors, between vector and the matrix, right? Same thing here for our continual classification problem this week. We will still use quite a lot of those dot products between vectors. And you know what this dot product indicates. You can have 2 vectors pointed to different directions. And the dot product between them, first determined by how long each vector it is. They are, okay, the vector. Then determined by the cosine of theta between the angle of these 2. So if you ignore the length, so you normalize your vector, and these two norms become one, and you only care about the angle between these two vectors and tell you the relationship between these two vectors and their data points they represented for, okay? And you also know this, that's your homework. You know the product of these two, just assume B has not been normalized, and you have to normalize B by the length of the B vector. But the dot product between these two are the A projected on B. So how you can compute the length of the projection. We also learned that PCA is to rotate your data points in order to let the new access to see more the variability among the data. And LDA is the same as the PCA. The difference is LDA deals with two different classes of the data points rather than just treat them as one group. So LDA will found the first PC, the principal component of this vector, the green one, in order to separate two groups of points rather than to display the overall variability between all the points, but rather, in this case, want to display the separation between points of red and blue circle. Okay? So this is like a very quick review of previous what we learned. Well, when you do LDA, you assume those data points are generated from some kind of distribution. You can use a single lines to capture its variation. Well, this so-called generative algorithm, because you have assumption the data was generated, follow some distribution which you can learn from the distribution. You can learn the variance in the data points which generated from the distribution. Well, SVM stands for support vector machine. This is a very different, it's not a care. This algorithm doesn't care the distribution underlying the generation of data points. It only cares how it can use the information to separate two groups. So it's called discriminative tool or model. It's more straightforward, practically straightforward to find the place which you can separate two group of data points, not assume any population distributions. So there's no assumption of distribution. It's binary classifiers. It is first separate points into positive, negative. So positive belongs to one group, negative belongs to different group. Okay. It focuses on the points which in the boundary of your separation, and not care about those points which very far away from the. So let's do this by two different colors. Okay. So if you have a group points here. Let's see, there's central node somewhere here, this one, and then the right population in here, okay? SVM will only consider the closed points we So this is one of the important ones for SVM. This is another. Okay. This is called the support vector. Okay. Vector is a data point, right, to support the separation of this. So that's what SVMs target for. This is a very small example to show some operation of linear algebra can build a very simple classifiers. Only use the dot product. All right. So, so this small example is like this, or like this. So we have two group of points. In this case, a red one and a green one. Then we found the central point of the two of them. So let's see, this is C1, C plus, C plus, blue one. Okay. And C minus is the red one. So it's two central points. This is in the space of some space. So you know this is a C1 vector. This is C2. So you can have two vectors. Then we are going to look at the difference between C1 and C2 first. So this gives you the vector W, okay? So this is W, which is the difference between C1 and C2. C plus and C. Then we found the central point of the C1, C2. Right here. So this is C, okay? C plus, C minus. That's central. Then we are going to have new data points comes in, and this data point. So let's put a hypothetic perpendicular line in across this W, which across C and orthogonal to W. So now we have a new point. So this is like X, okay? So once we have a C and a W, this classifier is already built on the information which we know from the two distribution of those data. Once X comes in, and we are going to compute a value. This value, so the X is a vector as well. So you can see this is a vector X. What we compute? We will compute dot product between X minus C, okay? So we compute, once this new data point comes in, we compute X minus C. This is already known. X minus C is this vector, okay? We do a dot product between this and the W. That's it. So we use this dot product to indicate whether this X is biased towards this direction or that direction. So this dot product gave you the information of the theta, basically. cosine theta value. So if you 90 degree against this difference between c plus, c minus, and this becomes 0. If you have a data point which in the middle of this line, there's no separation. But if you shift or bias towards this direction, and your cosine become like positive, and then you belong to this group. If cosine is negative, it belongs to this group. So it's only using the sign of this dot product to indicate which direction, which class this new point should be classified into group. Okay? So this is a very lightweight classifiers that's only involved in compute dot products between the difference between this and also this already pre-compute. So this costs nothing. So very lightweighted classifier. So this example just indicate to show you some of these combinations of the lineage algebra operation can be used very easily to build these classifiers. Well, the support vector machine is more sophisticated than this. Okay, this is a very naive, simple one. So this has been developed for a long time. We have a timeline of the machine learning algorithm development, remember. The neuron, the framework, the idea had been developed, invented in 1943, okay? People start to think about this. This is even before this ENIAC machine had been built. Then 1963 support vector network, the idea behind those support vector machine had been proposed. And then after several years, so this paper is 1995, same author with his students maybe, published this support vector networks, and this outlined the framework of a support vector machine. As I said, this machine algorithm consider those vectors, those data points, which in the interface of the separation between groups of data, you want to separate it. Oh, wrong button. Remember, so for machine learning, the kernel method and the support vector can be expanded into a whole semesters of teaching. So if you look at this as a kernel method in machine learning, this video playlist from a whole semesters of teaching involved all different aspects of the kernel method in support vector machine. There are also technique documentations we can use to learn this. So I compress all the lectures into this 1 hour. So you see the information is really condensed. So if you look at this user guide support to the support vector machine, this paper, you will find the figures like this, and you also found equations looks like this. So this is dot product between vectors. Okay? So I will explain why their equations look like this appeared in support vector machine, why, what they are computing for. See this? Okay, this is called a discriminative function. So we have a discriminative function, which is this times that, this descriptive function to determine the membership of these new data points in this machine. This is a small machine. Okay, so all this. Yeah, I just want you to be aware this is a highly condensed lecture, and to get the top level understanding of the concepts rather than many details. Okay. Otherwise, it take a semester to learn details. Okay, we only care of the bigger pictures. So here the big pictures is this binary, a linear binary classification problem is support vector want to solve. So if you want to separate those green cross away from the red circle, you can, in this 2D case, you can build several separation lines. Those are the functions of the discriminative functions. If you have the points which located to the left of this separation line and this one group, if you have points on the right side of this, it's another group. If you have a high dimension, these lines become the planes. So this is called hyperplanes for the higher dimensional data points separation. So the idea is we draw a line first. So you know how to define a straight line. So remember, if you have function, remember this. Okay, you have a straight line, something like this, right? You know you can write how to write this straight line. Y is some slope x, so alpha x plus a plus b, okay, so a. Right, you know how to write the function to represent the straight line here. So you can describe this straight line with two parameters, okay? Use this and that. So for us, we are going to find these two numbers to build a straight line somewhere here. So in this case, the slope is negative, right? In this particular example. So the alpha should be some negative values, okay? So some negative values, right, to indicate the slope. And you have intercept here, just how long this b is. Okay, so this is the basic concept. We need to learn some parameters to define a straight line in this 2D space. And this a and b should be selected. Allow the separation of these two groups as clear as possible. So what that means is clear, is we have to make the closed points of each group to the line, the close to the line, okay? Which is these two green dots and one red dots. We need these three dots which close to the separation line to be largest to the line, okay? First, we need to find those points which close to each other. So we need to compute the distance. Then we need to compute the distance from points to this separation line. And we change different A and B to adjust slope and also intercept until the distance from those separation, which the points in the boundary to the line as big as possible. The big means the buffer between buffer zone between the two separation is large. So we want to maximize the margin. Maximize, make it bigger as possible. So this involves a lot of the calculation from data points to the straight line. So I need to explain to you how can you compute the distance from a given point, x0, y0, to a line which indicated by an equation like this, okay? So this equation is it's also the same, okay? Basically, it's the same as this. So we understand this, but this normally the equation we can write into ax plus by plus c0. You can do the transformation to make this happen, which you put y somewhere in this side and then do some rearrangement. It becomes should be let's see whether it's correct. C3. So these are the same, right? So A and B, and this is a slope. This is a constant which just rescaled a little bit. So just make sure this is the straight line, this is a straight line. This can be easily explained by the slope and intercept, which this is ABC representation, but they are same, right? So I want to show you if we have a system like this, ABC, then we have a given points, say x, the point here, the P, okay? If you have a P, x, 0, y, 0. So if we have this point in the space, then we have this function of the straight line defined by the three numbers. There very simple equation can compute the distance from this to that, okay? This equation is so simple, so make the overall computation from data points to the straight line very easy. Why we need that such easy computation? Because we want to find a good separation line, so we need to try a lot of different combinations of the definition of these parameters to give you many different definitions of this separation line. So we need a quick computation to allow you to compute the distance from data points to straight line as quick as possible. Because once you change this and to a different angle or different slope, you need to recompute the distance again. So we want to accelerate this, so simple computation will be beneficial to the overall optimization speed. So we try to find this very quickly. Okay. So this example, I will show you at the end, the distance you can compute is this, okay? The distance from a straight line to, from the P to this straight line will be so the distance at the end will be just this operation. So in this case, this is the P, the value of vector. So we have a W times P, so in this case it's a P. And then minus C normalized by W. Okay, so from this equation you can see the P is the point, which is this vector. Okay, so every point in here we have x, y, so it's one of the points. So this point has do a dot product between this point with a W, which W is defined by these two numbers A and B in some way, okay? So this is a simple dot product, very easy to compute. Even though you have many of them, you can compute just by all the points put together become matrix, then multiply it by one W, which is defined by A and B. So this is very easy to compute. Then you compute all the distance. So this is for all the distance, all the points. And then you minus C. C is a constant. So minus C, nothing, okay, compute. And then you normalize by the norm of this vector. So the whole computation, this compute all the points, the distance to a straight line here becomes dot product. Basically, that's all, right? So this computation can be accelerated, okay, it's optimized in terms of speed. So that's why I want to spend the time to indicate, to illustrate why this computation is the distance from the data points to a straight line. So if you know this is true, you know, okay, all the computation in this have been reduced or compressed into a dot product between your points to this W, okay? So now I just want to show you what's going on, why this is the case, okay? So in this example, I use a straight line, which is, let's say a straight line is y 5 over 3x plus 5. Okay, same as this equation, is just we use a real number to replace A and B. Replace A and B. Okay, so this equals to 5x minus 3y plus 15. These are the two same line, right? Just this becomes 5, and this is -3. This becomes 15. So this is our example. So this have an indication. This is slope, the intercept. So 1, 2, 3, 4, 5. So that's 5, and then you have 3 over here. Not so accurate, but this is a line. Okay. This is what we define. So we're going to use one of the point, which is here, as an example. So this is a x. This x1, y1, x1. It doesn't matter where it is, but we use this as an example. We are going to compute the distance from this point to this line here. So this distance we want to compute this. So first, I want to find a vector which is this w. W is 5 and -3. So this go 3. So these two numbers are from A and B. Okay? So this gave you the idea of this vector. Okay? So this vector will be also going to go to this line. These two numbers is related to the slope. And if you compute, this will be just perpendicular to this line here. Okay. So the distance between this will be then I will draw a straight line here to indicate the vector of this x. Then I will extend this to make it longer. And the green line, this green line, maybe I should make it using this pink. This is your point, right? This is the vector of your point. So you have this, the point you want to compute this point to this straight line, the distance. So first I will project x onto this vector. So this projection gave you the how long this is. So this is also, say, the D, okay? So you can project this onto W. So this will be this times W minus. Make sense, this? Okay. So I know how long this is. And in order to compute small d, I just need to minus this part. Right? So this part is give you another value. See, this part is let's see, third. So I need to have this distance to minus the third. How long this part is to give you the value of how long the d is. In order to compute this distance, we found this point. It's located on this straight line here. And because of that, this is called V, and I gave the value, which is the V. So this V is xy, okay? So this V, in order to compute this distance, we need to have V times W. We project, no, not project. I think this is V. So this V is on the curve. So now we know, for example, it's x and y. It doesn't matter where it is, but we know it's on this straight line. So we can compute its projection onto it by using V times W and divided by V norm. And because, so this is D. And because this V and W multiplication, you can do this, and VC plus C equals 0. So this is on the straight line, so this whole thing is minus C. It's negative C divided by W. So this is x, the y. It's on the straight line, so you have x, y. So at the end, the dot product equals to negative C. You can put that in the equation to get this. So at the end, we have the equation from everything here. We derived equation. The distance of this point, x, is this. So V is this point. V times this. This is the expression of everything. If you expand this, it becomes 5x minus 3y plus C equals 0. So can push this back. This becomes C, okay? Minus plus. This becomes C. Think of this C instead of minus C. So the one I'm highlighting using the laser here, so this part is this. W times V is C, okay? Because V is on the straight line. So this gave you the equation of the distance, compute the distance. And this use dot product and also the relationship between this vector and this point is on this straight line. So once we know how to compute the distance to the straight line, and we can start to compute a lot. We compute all the points to the line. So using this equation, look at this equation. So the W is defined by this, okay? W is here. And that's your data point, X. So if you want to do some transformation there, a function phi, if you don't do anything, you can do the in itself. So this phi, this function is transformation to make the original data in the D space, dimension big D, into something else. So if you want to do this kernel transformation, you can do. If you don't, you just take original X, you can compute the distance directly from the original space, okay? The data points to the straight line. So what we want to do, we compute all the distance from the right points to the straight line and make this distance maximized, okay? So make them separatable. So this is at the end become optimization problem. So you see here there are max and the minimum, okay? So the first step is to use a minimum to find the points which close to the straight line. First find the points using minimum function to find the straight line which neighbor points close to it. Then we change the parameter of the straight line definition to maximize those selected minimum distance points to make this distance as large as possible. So the first step is to select support vector points. The next step is to maximize the separation by changing the straight line's direction or parameters. So that's why this function of optimization target function involves both minimum and maximum. Minimum is to select points nearest the neighbors, straight line, and then maximize the separation, the margin between them. Okay, so the margin you can compute using this expression after all the parameters have been rescaled. So you can do some transformation to make this function work, and also the expression of the margin, the width of the margin is just 2 over the norm of this W. So W is the slope basically, okay? The two numbers in the slope 5, -3. So you can change the slope by changing those two numbers, right? And try to keep this as big as possible. The problem is sometimes we cannot find the optimal solution. There are always some error points. So for example, you tried the best, but still these points are located within the group of red ones. And the red point of this as outlier is located inside here. So we need a soft margin SVM to tolerate some levels of mistakes, even though you have misclassification, but still these are the best solution we can have. So there are some compromised, balanced cutoff, which discriminative line we should at the end to achieve. In this case, for example, if you want to find the absolute solution with hard margin, and you should have the separation, which is this red dashed line. But because of this, take into account the margin, how wide the margin is, you should use this green line as a separation. Although there's one mistake, misclassification, but gave you the much wider buffer zone, and this classifier become more robust, because the next time if you have a real data, when you have applications, this will give you the best separation, will tolerate a few outliers. But this one is just too narrow, and you can make a lot of mistakes in small shift in the location of new points introduced in. So there are some of this compromised term which allow you to take some of the mistakes into the likelihood the loss function. Okay, so this is called a softer margin SVM. So if you emphasize this a lot, then you can have different loss function. You will end up with a slightly different selection of this separation margin, and depends on how much you want to emphasize this tolerance. So now we will consider what happens. So you can use soft margin, but sometimes if you have the problem like this, and they're just not separable. In this case, we want to introduce this transformation, the kernel function. Okay? So you just not be able to separate using linear or even tolerant to the mistakes. You cannot do anything with this linear, non-separable data. It's hard to find the straight line to achieve the good results, whatever you want to use. So this kernel transformation is to help you to push your points into a new space. Okay, in that new space, they become separable. Then you define a linear separation using SVM in this high-dimensional space, a different space dimension. In this example, we make 2D data points become 3D. And then suddenly they become separable using linear combinations of these planes. So this is the idea about expand your dimension rather than reduce. People always think we need to reduce the dimensionality in order to understand better of the data. So we have a PCA, we have a lot of other TSNE. They all try to reduce the dimension to look at the data. But in this application, we might want to make a lower dimensional data into a high dimensional space. Why? To make them more separatable. This particular example is you have 4 data points. Okay, this is a -1, -1, which green one, and +1, +1, this 2. Okay, and then you have 1, -1, -1, 1. So in this 2D space, if you introduce one straight line, you just not be able to separate them all together. So what we can do is to increase dimension by introduce the product of x, y as your third dimension. And then once you multiplicate x, y for each point, and these 2 right points have the third dimension as a negative value, which these 2 positive, these green dots, 2 green dots become positive value. So you can introduce a plant which in between positive and negative, which cross 0, to separate these 4 points perfectly. So that's the idea about expanded dimension to transfer original space data to a new space, maybe higher. Okay, normally it will be higher, through this feature engineering. Okay. So the feature means these data points are defined by 2 variables, x and y. Now we synthesize a new feature, which is z. Z is the product of x and y, and introduce these new features in the data points and make them more separable. So those are the whole idea about kernel method. There are many different kernel functions you can use to transform your data. Okay. So the kernel function is to use some of these combinations of the original variables to transform original feature space into through the kernel operation to transform the kernel feature space to make a data point separable. This is another example. This is like 2D example. You cannot separate red from the blue points. But you introduce the square of x1, and then they become a quadratic feature. In this space, you can have a straight line to separate them. One of the functions we use very commonly is called a Gaussian kernel. So this is a normal distribution curve, right? This is like 2D. Then you found a central. Okay, central is z, which may be located in the middle of your point. Then you compute the distance of your original data to the center. If they're close, then you translate into the high probability, which is in the center, and you have a higher y position, z position. If they're far away from the center, then you make them lower. So you, at the end, you create this 2D Gaussian surface to map your original data into the high dimensional space after you introduce this Gaussian kernel. So this cartoon illustrates the process. Those blue points we want to separate from the red points. So we found the center, and then start to introduce. If they're far away from the center, then I gave them higher value. If lower, I keep them lower. So then introduce a plan to separate them. Okay, so this is summarize the input sample space versus kernel feature space. The same as what I just described. The whole purpose is make non-separable, non-linear relationship among data and become separable in the higher dimensional space. Okay. I think we can take a 5 minutes break before we move on to cell-cell communication study. Okay, so in summary, in the machine learning field, kernel methods are a class of algorithms for pattern analysis, whose best known member of the support vector machine. The general task of this is to find and study general type of relationship in the data sets. Kernel method named from the kernel function. Okay, so this is transform your data, enable them to operate in a high-dimensional space, implicit feature space without even computing the coordinates of data in that space. So, so there's a term called a kernel trick. So this is the dot products between your point after transformation. The kernel are the same as original. It takes some time to digest. So it's just a very quick way to compute, to allow you to select the correct kernel function in order to minimize the computation. There are many different ways to select this kernel function. But if we select carefully enough, then the dot products between points in the lower dimensional space will be the same as dot product after you transform or using the kernel function to transform into the high dimensional space. This gives you some advantage in reduce the computation, because in lower space, we need a lot of dot products to compute the distance between points, for example. If their computation is the same after you transform into the higher dimensional space, the dot product keep the same. So we don't have to really increase the dimension of the data. We just directly compute the dot products between data points in the lower dimensional space to reflect their relationship in the high dimensional. Then the dot products help you to compute the separation. This is one of the examples. If you look at this example carefully, this kernel function is something like this. Okay, then the dot product after you transform x, y will be the same as you do that before, okay? Before the transformation. So basically that's the expression. So the kernel trick is to help you to accelerate the computation by carefully select the kernel function, allow you to accelerate your computation of the SVM method. So the kernel method also apply into traditional PCA. So if you do the transformation, after transformation, then do the PCA, you can separate those points, okay? Without the transformation, you just cannot rotate your data points to separate blue points from the red one. But if you do this kernel transformation, transform your data, and then do traditional PCA on the transformed data, they become separatable. We will place some of this data. We will synthesize a points which in the center of this cloud as one cluster, and then the blue one is a circle around it. So traditional PCA, you just not be able to separate them. But if you do the transformation, see the kernel function, we will define the kernel function to change your data, and then after kernel function transform data, then your PCA will be able to separate. So we will play this in the computer session. Okay, so in the last 45 minutes, I will introduce cell-cell communication or cell-cell interaction analysis to give you examples and introduce the principle behind that. So in biology, we always compute statistics called heterozygosity. Sometimes use a big H to indicate a number. Basically, tell you how, if you have a population of animals, for example, you want to see, okay, how much genetic variation or heterozygosity among the population. You need to compute values for it. So in that case, that's some concept in the population genetics. So we compute the allele frequency from individual SNPs. Okay, SNP stands for single nucleotide polymorphism. So if you have a SNP, see, okay, the location in the genome, 50% of animal have A, and another 50% of animal have a C in this location. So this is a SNP, and this is two alleles, and to indicate the genetic difference between populations. So if you use one SNP to compute the heterozygosity, this 50-50, the frequency will be the largest heterozygosity you can get. This is a more heterogeneous population. Then you have 50% of animal with A allele in this position, another 50 with the C allele. Okay. So this is indicate the idea. So if you have a population with PQ indicate two alleles of the A and the C, for example, then the product of the P and the Q gave you the largest heterozygosity definition or for the whole population. So if you treat this as a group of animals, then this is the highest heterozygosity level you can get from compute this biallelic SNP. Well, if you have a population look like this, 90% of them have a P, so 90% A and 10% C. This is a low heterozygosity population. Low heterozygosity means more homogeneous. So you're relatively more homogeneous. So the definition of heterozygosity is that you have the chance, the probability, if you pick two individuals from population, how likely they are going to be differ. Okay. In this case, if you randomly pick two, and they most likely to be two white, okay, because your P has a higher frequency overall, so the more homogeneous this population. So that's why you can use P and Q, the product, to compute the likelihood that you can get the difference between randomly pick two. So this is the same idea. So 2PQ, or remember Q is 1 minus P, okay? P, and the Q is 1 minus P. So the product of PQ is the heterozygosity. So everybody get this, right? No problem. So I want to give you this idea is because when we talk about cell-cell interactions, we want to measure how strong the interaction could be. Then you need 50% of this, 50% of that to cell type in order to express, in order to communicate more, right? So same idea, we need to have a P times Q in order to indicate how likely the two cell type will be interact. If you have 90% of cell type A, 10% of B, you don't have strong interaction compared to 50-50. As I said, so single cell data analysis gave us a chance to study this by looking at potential interaction between different cell types. Of course, we already dissociated cells into separate groups. So what we can do is to just infer from the data. At the end, we look at the matrix of this expression and separate the cells into different cell types and to infer the likely possibilities between interactions through ligand receptors gene expression. We remember nowadays we also have a spatial transcriptome data which gave you the location of the cells. So if you know spatial transcriptome, it will have the single cell level and also the different cell type. You know where the location is. You have a lot of cells in this place in green cell type, where in red one you have a red cell type. Then you know the interface between them. If you have that information, if you know some of these gene expression pairs, then you can infer the relationship between interaction. This give you the better resolution compared to only you have dissociated cell population. It's a very important topic because we understand the communication between different cell types. The communication is so important to give the signal, for example, cell differentiation development, give you the interaction between signaling to keep tissue homeostasis functionally. Most importantly in immunity study, so you have immune cells communicate to each other, form networks, and also communicate with directly with other cell types. There are different ways to do the communication. So mostly we will discuss this mechanism called the pyrocrine. It's between neighbor cells. One cell gives the peptide product ligand, okay? Could be the protein and secrete into the extracellular space. Then another cell type using the receptor which specifically recognize the ligand produced by another cell type to communicate through this ligand receptor mediated communication pathways. Cells can do autocrine. That means the same cell type produce ligand and gives feedback to its own receptor to initiate some of this positive feedback to itself, maybe negative, I don't know, the self control. And you have this juxtacrine, then you have to physically contact to each other and using this channel to transfer ligand from one to another. So this is like physical contact between cells. And you can also have long distance endocrine pathways. You can secrete some hormone peptides, and then the information will pass on through the bloodstream and to carry the information to the long distance other place and to have the endocrine effects on the cell type which have a receptor for those endocrine signals.
So we will focus mostly on the pyrocran here, this mechanism. If you have a single cell data which collected from different organs, maybe we can study endocrine, and if you know some of those relationships, but I haven't seen much of those. So maybe it's a good active research field. We can study single cell data from different organs if you believe their endocrine mechanism to help the communication between two cell types, between two distance related organs. Over the years, there are many tools has been developed for single cell communications analysis. There are different names. Cell chat will be one of the major ones, and they all based on the idea about the compute the product between P and Q. Okay, basically, you have the expression level of ligand. You will multiplicate these values with, so this is the expression level of ligand. You will multiplicate by expression level of receptor. So almost all the, let's see, so this communication so gave you a value L. So almost all these methods based on the idea of this. This is one cell type. This is gene, ligand. This is another cell type. And we look at the gene of receptor. From our biology and knowledge, we know this is a pair. This is L, R. This is a pair. We know they communicate. We have some other evidence. They are not a random gene expressed in a coordinated way, but there are some support from biology. So all this method is based on this idea. So you can do this after you have cell type annotation. So you have to do the basics. You have a filter of the quality. You have a dimensionality reduction. You have a cluster of cells. Then you have annotation of cell types. So after that, we start to compute the average expression of genes. So you compute average expression of genes in different cell types. Remember, this is green, this is pink. So there are different cell types. Then you compute all the genes expression. Then you found ligand genes from one cell type and look at its receptor gene in another cell type. You don't look at it itself. If you look at the ligand receptor gene in the cells, you are studying the autocrine so by itself. But in this case, we want to look at the two different cell types. Then you compute for the corresponding ligand gene with the corresponding receptor gene. Look at their expression, just do a product, and then to get this interaction score, okay? So this interaction score sounds awesome, but it's just a product of two expressions. You may want to do some normalization of some, but in general, this just indicates how significant of this relationship is. Okay, if you have 9 and 1, your product is 9. But if you have 5, 5, your product is 25. This is higher. 25, it means there are higher interaction score and the most significant. So where you can find the database of ligand receptor. This is an example. The database is called Cell Linker, and this is just like a review paper. The authors look at the different literatures to summarize and found ligand receptor reported from other studies and summarized into a database. So you can, for example, select human, then you put the gene name. So in this case, EGFR. So this is a receptor gene. The database will search, okay, for this receptor, what other genes are the corresponding ligand genes? Ligand genes will be the other cell type produce ligand and communicate, it binds to this receptor. You can download the whole database from download this TXT file. If you look at this carefully, the ligand ID is this NCBI gene number. And then ligand symbol is the gene names. And then you have different types of secret or membrane. Then you have, for the same row, each row you have a corresponding receptor gene ID and the symbol and how they communicate in what way. This is a many-to-many relationship database. So you can have one ligand have multiple different receptors, or one receptors may have multiple different ligand. Okay, some ligand may serve as both ligand. I don't know whether some genes will be served both ligand and receptor. Maybe not. I don't know. It's complicated, but this is a pre-knowledge you can use to help you to calculate this. Okay. But maybe you have a question, if I have some unknown ligand receptor information, it's not been collected into the database, you certainly will miss that if you use these tools. Those tools are based on this. Okay? This is one of the problems, so we probably will miss a lot of those unknown ligand receptor relationship if you only rely on pre-knowledge, and if they haven't updated yet, maybe several years ago was okay, but not no. Okay, so this illustration to show you two cell types, cell type A and cell type B. And for the ligand 1, so this is one pair, ligand 1, the receptor 1. Cell type A has expression of 3, and cell type B has a receptor expression 7. So we have an interaction score 21. That's easy, right? So if you have a condition looks like this, in this type, the score is still 21, but it's different direction. So it's from, in this case, the communication is from the cell type A to B, because the cell type A produces ligand, B produces receptor. Well, this is opposite. Will go that direction. So this analysis you have to consider both ways if you want. Okay, two cell types. Who's the donor? Who's the recipient? Who's produce the receptors? Well, if you have a condition looks like this, cell type receptor A express level 3. Well, cell type B also have the receptor 7, but this is not a pair of the, then you don't have this value. If the ligand expressed in both cell type but no receptor, so regarding this pair of ligand receptor and then this cell type, these two has no communication between these two cell types through this this pair, okay? No communication. So this is a real example from NCBI geodatabase. This is accession number, and this is a mouse skin tissue wounded. Then we can download the data by using this accession number, and then to find the matrix, the gene, and the cell barcode. So this single cell data, we can load the data into MATLAB, and this is a pre-processed data, contains 28,000 genes and 22,000 cells. That's a huge data set from one animal. Then reduce the dimension, right? You could get this 3D Disney plot. We focus on this and start to annotate the cell type. And we found a few different cell types. They are fibroblasts, okay? Remember the skin cell, and then it's wounded, and then start to have immune cells or endothelium cells around, and they're stellate cells, okay? This is from the paper. I believe the author had the paper and then published their data. They explained their communications between different cell types, stellate cells also get involved. So this fibrogenic process and to help to grow. So what we can do is to select two cell types to focus on, okay? So this is pairwise. Then for this example, I select a pair of the ligand receptor. So we focus on two cell types, then look at this receptor and this ligand pair. So this is from the database. And this is important to involve in the wound recovery and the growth factor receptor signaling related. So that's why I select them to demonstrate the expression of these two genes in two different cell types in that example data. So this PDGFRA is the receptor. So PDGF receptor here. And this should be the ligand. The two cell types, we focused on stellate cell and endothelium cell. So you can look at all of them. You can build a interaction score, okay, from one direction to another. That's what we found is strong from the endothelium cell to stellate cell. You know how to compute this score just by looking at the product, the values, right? The average expression of those two genes in each of these cell type. So this network shows 3 cell types. I ignore the macrophages. The direction has been indicated with the arrow here. and the value is proportional to the width of this linker. Okay, so the strong link between endothelium cell to stellate cells has been showing with this thicker link from that direction to this. It's different from over, so they're not symmetric. It's one direction, strong, but not the opposite. Then we look at the gene expression of the CAV1. If you look at this density, and also if we have a color bar, it should be higher in endothelium cell. So basically this indicates CAV1 relatively highly expressed in the endothelium cell. We'll look at the receptor gene. So this is a different gene name. It should be PDGFRB. This gene expressed relatively high in stellate cell. Okay, that's why the score for the communication. That's why this link becomes stronger, because the corresponding gene expression are strong. Their product is a higher average product, which the other way around, this from this endothelium cell to stellate cell is not so strong. Okay. This is just a different view, the same information, and using this bar to indicate how high the expression level is for individual cells. So you can see these peaks are relatively high, and also the density is higher in this figure. And correspondingly, this PDGFRB, the receptor, highly expressed in the stellate cell. That's why the indication is strong for this pair. As I said, so all the algorithms use the information of this. Very simple. They just color-coded in different ways, and to make a package, to claim they're good. But the algorithmically, there's no innovation. Basically, people just use the same kind of information over again. Some tools say we use a larger database. Some tools say, oh, we synthesize, we aggregate different pairs because you can compute one pair of ligand receptor, you can consider another related, you can aggregate, make a comprehensive interaction. Some tools say, oh, we can do multi-ways of communication, put them into pathways, but essentially there's no much difference. They all rely on this product. Because of that, my lab and my students have been developing a new tool called the single cell 24 XCT. XCT stands for interaction. So we believe this pipeline is the best in the market, although not everybody using it. It's not as popular as others, but we have a lot of creations in the algorithm design here, and not only rely on this, but also consider networks, also consider the variability. So if you have a gene expressed very noisily across the cell, and we will take that into account, because those genes which the average may be really high, but because the noise indicate they are very active. And here also, if you have a gene expressed across cells very differently, so we take that into account when we compute the score. which score is stored in this matrix. And this matrix contains pairs of gene and gene pairs I and J, and I and J are the different genes. They are ligand receptors. We consider different cell types, and also for the cell type itself, we build the gene regulatory networks. Okay, so this is one network, and this is another network, and this is another network. So 3 networks. They are matrices. Okay, we put them together. Okay, put them into a block matrix. Let's see, illustrate the block matrix. 3 matrix, build a block matrix. I just want to show you how the information gets synthesized, aggregated to help identify these interactions. So you know we can use matrix to represent the network. So we have first cell type 1. We build a network which this is gene by gene. This is one matrix. And then for cell type 2, it communicates with, we build another matrix. It's also G by G, okay? Same dimension, same number. Then we complete this block matrix using this 2 off-diagonal position, and this is a S matrix here. Okay, and this transposed S. So this is a symmetric, basically. So what's inside S? S indicate the ligand receptor communication scores. So if you have a gene, so this gene here is a ligand gene. So L. And this L will have been here, and then this location will come back to its receptor. So this is receptor gene. So this coordinates here indicate ligand gene and the receptor gene. So L, G, so I, J. So it's just a pair of genes, receptor. So you can do all the combinations of all the ligand receptor you know, you fill up this matrix. By looking at the ligand genes in this cell type, receptor genes in this cell type. So you can fill up those correspondence matrix. So the whole thing become a block matrix. Will be put into a neural network to compress the dimension. So here, if you look at the rows here, the first G, first set of G, so those set of genes from cell type 1, and then the rest, so G, G1, G2, the second half of this block matrix are the information from genes from cell type 2. And then those will be reduced into a lower dimension. Say if you have a 3D, then all the genes will be pushed into here, and some same set of the cell type 1 genes and the same type of cell 2 genes. they all compressed into a low-dimensional space. Then we want to compute the distance between one gene from cell type to another gene from the receptor from another cell type. So we look at the distance between ligand receptor genes in this reduced dimensional space. If they are close to each other, we believe they have stronger interaction. So we use this distance-based method to find the gene ligand receptor pairs. Why this is useful? Because the information between genes in relationship has been learned from the network itself, and also corresponding matrix. So all the information has been synthesized into this. So we found the gene ligand receptor pairs and which ones are mostly close to each other in the reduced dimensional space to report them. Okay. When we build this, we don't rely on ligand receptor pairs which already know from the database. We can have unsupervised or no prior lineage, no prior knowledge information, so you can compute score for all the combinations of genes. In that case, we can develop the method which develop a new ligand receptor detection, right? Because this is the data driving without the information of whether it's known ligand, known receptor. As long as you have a higher values in this corresponding space, you have a chance to be selected as a ligand receptor pairs at the end when they report the results. But you also need to have a support from other genes from network to make. Some maybe have very strong correspondence, but not supported by the network itself. Some weak interactions may be supported by the network, we will still be able to find them. So this is a workflow. We found annotated cell types, then build a network for each of them, then to learn the correspondence matrix, that's crosstalk scores. And then in this equation, we introduced variation, okay? So you have average gene expression and also take into account the variation of that gene expression in the computation of the score of this. Then there are dimensionality reduction, alignment, okay, to make them. This is an example applications to find the gene interaction between fibroblast, inflammatory, with dendritic cell. Okay, those are the ligand receptor pairs. And we can find ligand receptor pairs. We can make a network, and those gray genes are within the cell type other genes which closely related with those ligand receptor genes. So you can see a lot of support for the network to enhance the communications between these two cell types through these ligand receptor pairs. These two pairs at the same time we illustrate. This is like three members, three different cell types involved in T cells as well. That's interaction between inflammatory dendritic cell with T cells through those ligand receptor pairs which we are known. This block matrix A is what I just described here. We designed this. This can be further to introduce another block matrix. This is from cell sample 1, and this from sample 2. Sample 1 means you want to compare with sample 2. And the sample 1, you have 2 cell types. You want to study the communications. In sample 2, you also want to study communications. And then you want to see the same communications, whether they become strong or become weaker in different samples. So this is a framework to compare 2 samples. The matrix become bigger. And we can see, okay, this will be a healthy individual sample, and this will be from patient, okay? And then you can detect some communications between pairs and become stronger. Some become weaker in the disease, in the patient group. So those are the application of 2 samples. We are also trying to use the spatial transcriptome information here. And then, for example, this will be the cancer cells, solid cancer tissue. And those are the stromal cells with infiltrated immune cells. We want to see if the cells of immune cells close to the boundary and start to fight against the cancer, what are the expression, what's the interaction change between them compared to which are not have been registered as a front line of fighter. So we can compare the interaction between neighbor cells against non-neighbor cells to see what kind of interaction had been enhanced in the fighting between cell types. So those are some the recent progress we are trying to make. Okay, that's all for today. Let me know any questions if you have. Okay. No? Good. See you